is the cerebellar pathways that we talked about with Ian. You see the three layers. We'll be looking mostly at the Purkinje cells and how they connect to their output, which isn't shown in this slide, but it connects to the DCN, which is the deep cerebellar nuclei. We'll mention a little bit about Purkinje cells. They receive excitatory inputs from climbing into parallel fibers and inhibitory input from basket and stellate cells. They send their information to the DCN. They're GABAergic, so the information they transmit is inhibitory. And they also have very high intrinsic firing rates. If you just record from these cells, they'll be spiking at 50 hertz. And we'll be looking at synchrony, whether a bunch of Purkinje cells will exhibit synchrony if they'll be firing in a pattern. So these are the deep cerebellar nuclei. This is the human cerebellum. And you can see there are four nuclei in each hemisphere. So the dentate, which is the largest one, and moving more medially, you see the emboliform, the globose, and the fastigial. We're going to be recording from the DCN for this experiment, but the authors didn't specify which nucleus specifically. They gave depth and location, but not as much which of these four nuclei they'll be recording from. A little bit about the DCN. So they received some excitatory input from moss and climbing fibers, but that information, or our knowledge of that is that it's a weak excitatory information, if at all. And they receive inhibitory input from Purkinje cells. That's the majority of the input that we look at. And they send information to the brainstem, and it's important for proprioception and planning future movements. So we're going to be looking at whether DCN neurons respond to synchrony from Purkinje cell inputs, and we'll be talking about what that means in terms of modeling the input later. The authors say that there are a couple of factors that can contribute to synchrony. Among them is how many Purkinje cells are converging onto a DCN neuron, and they say around 40, and they'll be giving some explanations, they'll show some examples of why 40, where the number comes from, and they'll also look at the DCN neurons have high intrinsic firing rates, it's even higher than Purkinje cells, and they'll also be looking at the kinetics, about how quickly the cells can spike. And it's very quick, as you can see, it's 2.5 milliseconds. The animals we're going to choose are mice, and we'll be looking both at in vitro and in vivo. So what they're going to look at first is conductances, as a way of estimating how many Purkinje cells are converging onto one DCN neuron. So what they're going to do is they stimulated the axon of a Purkinje cell because it's very difficult to stimulate a Purkinje cell and then measure from the appropriate DCN cell. So they're going to measure from the axon instead. They'll stimulate the axon instead. So you can see in figure A, on the top you can see a trace. They're going to be stimulating in a declining fashion. You can see that on the bottom, 150 different stimuli decreasing, starting at 140 nanoamps. And when you're stimulating at the beginning, you always elicit a spike, you elicit a spike. And what they're going to do is they're going to use this information of the current to estimate the conductance for the DCN cell. So using the information that there's always spiking and that there's a certain amount of current, they determined that the conductance for this maximum stimulation is around 110 nanosiemens on average, and around 200 nanosiemens at its max.
Do you know, did they stimulate one axon or a bunch of axons?
I believe one.
Okay.
So they find the conductance for the cell for all these maximum stimulation, and then they move the amount of current they're injecting. They make that smaller and smaller, and the goal is to get to what you see on the left. That half the time you're getting a response, and half the time you're getting no response. Like quantal release, 50% success, and then at that point, if you look at the stimulus, it's around I think 15 to 20 microamps, and then they try to find the conductance for that, and they say that that's around on average 10 nanosiemens. So they find this ratio of the highest conductance, which is around 110 or 200 max, and it's 10 when there's even an event or there isn't an event. So what they conclude from that is you look at the ratio, that it's going to be around 12 or 10, 20 at its max. So what they say from that is that there must be 20 or 10 or 20 Purkinje cells synapsing onto this DCN cell. So that's why they say around it should be 20 Purkinje cells. But they also give themselves some wiggle room and they say, well, because of the splicing and because of other factors, that's probably a low-end estimate. So they say there should probably be around 30 to 50 Purkinje cells synapsing onto this one DCN cell. And they use other measures as well, which I won't get into here. They look at structural constraints and they look at some of the GABA A receptors, and they also conclude that 30 to 50 is a good estimate for how many Purkinje cells are synapsing onto this DCN cell. Okay, so some of the factors that we said contribute to this synchrony are convergence. We just talked a little bit about that. We're also going to talk a little bit about the intrinsic firing rates and the kinetics. So first about the intrinsic firing rates, they did this short experiment where they look at intrinsic firing rates in mice, this is in vivo, and they look at mature mice, or P17 to 26, 17 to 26 days after birth, and compared to more weanling mice, which are only 13 days, and they just measure the spontaneous firing rate in the DCN cells. And they show that it's 90 in mature animals and only 22 in immature animals or adolescent animals. So they conclude that the DCN intrinsic firing rate will increase with development. Apparently this was something that hadn't been done before, so this was important to show. So it's 90 hertz in the adult, and it increases with age, or up until 90 hertz. You can see that in figure A that the firing rate is much smaller than for P13 than for P21.
So is there a huge difference in maturation between P13 and P17 and a small difference between P17 and P26? Why did they choose these days?
They didn't specify why they chose that as a specific time window. I'm guessing it has to do with the maturation cycle of the mouse. I guess that 13 is, I haven't worked with mice myself, but I guess 13 is still in this really critical maturation phase, and then P17, 26 is it's already close enough to an adult that it doesn't matter. I can't speak to that. Yeah.
Did they say anything about what happens between P13 and P17? Is it like a gradual increase? I mean, it's 4 days and it's like more than 4 times the intrinsic firing. Did they say anything about that?
So they did. This wasn't the main focus of the paper. I don't think this was in the supplementary. They just kind of mentioned in the article and show the graphs and figures in the supplementary. So I don't think they really wanted to go into why there is a change or how the change happens, if it's gradual or sudden, and why it's over so short a time window, as you pointed out. I think what they're really interested in saying that this 90 hertz is really fast, and it's interesting, and I guess it had never been shown before that 90 hertz is not there through the entire lifetime, that it comes from the smaller value in adolescence.
So just in general, I mean, 26 is not an adult mouse. It's adult usually we start at 8 weeks to consider them as adults. But the rate that you get at 17 to 26 would be stabilized. So I mean, the mouse still continue to develop, but this parameter is not going to change anymore. So basically, these rates at 17 to 26 are just as adult. It's not an adult mouse, but the rate is like in an adult. And the other thing I wanted to point out, you see this on cell? What does it mean, this on cell recording? You see on the left-hand side on the bottom? It's just a technical question, kind of comment. So when you want to record the activity of a neuron, you can either record extracellularly or you can record intracellularly. If you record extracellularly with standard method, you're not sure that you're recording from one neuron on which neuron, blah blah. If you're recording intracellularly, you're going to change the cytoplasm of the neuron, so you don't know if what you have done is going to affect this property. So this is like an intermediate measurement where you go with an identify the neuron, you can actually electroporate it and put dye in the neuron at the end, but you're not going to disturb the firing property. So that's considered like the cleanest method to evaluate the firing property of the neuron.
Cool. Okay, so that's a little bit about the intrinsic firing rates of these neurons. They're high, and they also change until adulthood. And they're also going to look at the kinetics, or how fast the neuron takes to shoot again. So the authors talked about previous models that the time constant for the decay of the signal, like you can see in figure C, past models have been working with this assumption of 13.6 milliseconds. And what they showed is that, or they did a different measurement and they came up with 2.5 milliseconds. And the key difference is that they were measuring in slices that in the past they had been measuring at room temperature. But here they decided to measure at the artificial cerebrospinal fluid increased heated to close to physiological temperature, around 36 degrees. And what they showed is that the kinetics are drastically different. That the time it takes for the neuron to get ready to shoot again, to go back to baseline, is much shorter for body temperature than it is for room temperature. So the kinetics will play a key part in the synchrony in the DCN cells getting ready to synchronize each other. Question? Okay. So they're going to be using something called the dynamic clamp. I had never heard of the dynamic clamp before, but it's kind of a hybrid between recording and computers. You can do real-time calculations and inputting. This is an electrophysiology method that uses a real-time interface between one or several living cells and a computer analog device to simulate dynamic processes such as membrane or synaptic currents in living cells. And we're looking at the synapses, so this dynamic clamp will be useful. Okay, so we're going to be measuring synchronicity into the DCN cell as if it was coming from a bunch of different Purkinje cells. So using the dynamic clamp, we'll be delivering 8 trains of inhibitory potential using the dynamic clamp to the DCN cell. So the key is that these trains of current, potential, can either be asynchronous or synchronous. They didn't give us an example of what that looks like per se, but it's the issue at hand is whether the pulses are syncing up across these 8 trains or whether they are random and not matching up at all. So in figure A, you can see the recording from the cell. If you see the arrow, that's when the trains begin. So before that, you can see this really high firing rate at around 100 hertz or 90 hertz. And then at the time where the arrow is, you start with the asynchronous train, and you can see a really low firing rate. It's close, I don't know, maybe 10 or so. And you can see a reduced spiking activity over these 1.5 seconds. But then at the second arrow, you can see that it changes to a synchronous train that all 8 inputs are matching up, and you can see a much higher firing rate until the end of the stimulus, where it goes back to around 100 hertz. And you can see that we're stimulating with 50 hertz, and the neuron is responding at 50 hertz. So that's what we call entrainment. There's a train of the train that's being given in is also being transmitted. And you can see that the physiological time constant is important. That if the time constant is physiological of 2.5 milliseconds, you can see with circles that the synchronized activity has a high firing rate, but unsynchronized or desynchronized has no high firing rate. And if the cell kinetics are off and you're recording at the wrong temperature, that's how they vary these time constants, you can see that the synchronized activity goes down and that the desynchronized activity is close to or at 0. So the kinetics for the DCN cell are important for whether this synchrony can occur. Yeah.
Well, maybe this is more for Miki, but usually when we record from slices, is it at room temperature? Or?
It used to be for a long time. People used to work in room temperature.
Because this shows that it's completely different results.
It's true. So over the past, I think 15 years or so, it's more slices do not actually, you cannot maintain them for for the same amount of time when you do it in physiological temperature. They tend to degrade much faster. So, but usually it's almost standard today to record at physiological temperature. It's also technically not that complicated. It's just putting another heater on the flow that comes into the bath. But it's annoying because you have to all the time kind of calibrate it, but it's not very difficult. There's one point I wanted to highlight here. It's kind of strange if you think about it. What they do is actually they deliver inhibitory input, and they actually increase the firing rate. I mean, they don't increase, they actually decrease. The original firing rate is 90, but by stimulating with inhibitory inputs, they make the neuron fire every time they kind of deliver input. Did you pick that up? It's kind of strange. So you have an inhibitory input coming into the neuron, and every time after an input you get a spike. So you deliver the input at 50 Hz, and you get output at 50 Hz. Basically what happens is that, in between spikes there's an inhibition, and then when the neuron is recovering from the inhibition you get a spike, and then after the spike there's coming another inhibition, and it doesn't manage to get another spike in between these inputs.
So it's not immediately after the inhibition.
It's not
right?
on the inhibition.
Yeah.
It's not like the inhibition comes and then that doesn't make any sense. Right? So you have inhibition and then a spike. Inhibition, spike.
Okay. So the above we saw for a 50 hertz train, but you can also see a similar kind of train for other frequencies or higher frequencies. That if you stimulate, each triangle in figure C is a stimulation, and you can see that for the length of that is 1 second, or is 100 milliseconds. So if you project that, you would get for 60 hertz, you would get 60 spikes. Then we're going to compare the actual spikes with the dynamic spikes and see how those compare. So in this figure, we can see in the black circles, you can see evoked spikes, and then you can see that if the stimulation from the Purkinje cells is at around 60 hertz, then you're going to have a train at 60 hertz, 70 and 70, 80 and 80. But if you're inputting asynchronous information from the dynamic clamp, then the firing rate goes to 0. So what we learn about the DCN neuron firing rate is that it depends on two things, both the frequency of the input and also the synchrony of the input. So now we're going to look at another question. The authors say that synchrony is all well and good, but they don't think that all the neurons that are inputting, all the Purkinje cells that are inputting to the DCN cell are all synchronous at the same time. So the wonder is, what if not all of them are synchronized, but only some of them? What if there's only a subpopulation of synchrony? Will DCN output change as a result of this partial synchrony? So what they will do is that they use the dynamic clamp to input 40 IPSPs to this DCN cell, and they'll synchronize a fraction of them, either 0, 2, 4, 10, or 20 of these 40 inputs. They'll measure different firing rates that you can see up there. And the remainders of the ones that are not being synchronized will be asynchronous, and they'll have that at a constant 50 hertz regardless of which firing rate they choose. Okay, so the first thing they notice is that asynchronous activity will lower the firing rate because at least half of the inputs will be asynchronous, and that lowers the firing rate in general. So you can see there that all these have at least 50% asynchrony, so you can see that this reduced firing rate. This is for 25 hertz input. But what you can also see is that the more synchrony you have, the higher your firing rate. And it's around, for 50% at 25 hertz, you have around 13 spikes. So here you can see a raster plot. You can see the spikes lining up more synchrony, and you can see more spikes when you have higher synchrony. And this is the results for all 5 firing rates they tested. So you can see that for higher firing rates, you get, and for higher synchrony, for lower synchronized rates, you get higher firing rates in the DCN cells. One thing I wanted to point out is that when your frequency of the synchronized rate is too high, you don't get any spiking rate. You can see at 125 synchronized rates, the firing rate of the DCN cell is 0. It doesn't matter how synchronous they are. And I guess that's because if you have too much inhibition coming in, it doesn't matter how much synchrony there is. It's not going to get any activity from the DCN cell. And you can also notice that there's a lot of effect even if you have just 2 synchronized inputs. Only 5% of the inputs of this DCN cell, you still get some synchronized activity. On the right, you can see that there's a difference between between 5% and 0% synchronized. So even a little bit of synchrony can have a real effect on the DCN cell. So now one thing we're going to look at is the interspike intervals. So you can see here this is for 50% synchrony. We can see here also that we have to add 200 picoamperes of DC to compensate, because at higher frequencies you're not going to get any spiking activity, as we showed previously. And you can see that the intervals, the interspike intervals will cluster around the stimulus intervals. So, for example, in the top panel, you can see that if we're measuring at 40 hertz, that every 25 milliseconds you'll have a break. That'll be the next stimulus happening. And therefore, in the DCN, most of the interspike intervals will be either at 25 milliseconds or on multiples of that, 25, 50, or 75. You can see that for each of these different firing rates, that most of the interspike intervals will be clustered around these firing rate intervals. So you can see this is clustering around multiples of these intervals, as we said. So this is showing that there's phase locking of the DCN in response to input trains. That if the trains are coming regularly, then the interspike intervals will be clustering regularly at the intervals between the firing rates. So now we're going to look not only at the previous was all in vitro, and now we're going to look and see if this happens in an actual live animal. So we're going to stimulate the molecular layer, which is the top of the 3 layers in the cerebellum. And we're going to do that in order to synchronize the Purkinje cell activity. And we're going to record the DCN neurons in anesthetized mice. So this is an example trained at 40 hertz of what the DCN cell looks like. And on the right, you can see an image of what the cerebellum looks like. I wasn't really able to visualize what that is exactly, but there you go. And here you can see the recording from the DCN cell. You can see that in this case, the y-axis is the number of spikes and not the percent of the spikes where they're clustering. But you can see that for 40 hertz, that most spikes are happening 25 milliseconds, every 25 milliseconds, which is the interval of 40 hertz. Every 25 milliseconds you get an event. And you can see this happens for 40, 50, or 60 hertz. So now we're looking at phase locking in vivo. On the right, you can see the image I showed you before. That was in vitro. On the left, you can see the in vivo activity. So first, you can notice the baseline in red in the top of figure B. You can see that every 10 seconds there's an activity, which is consistent with that really high firing rate of around 90 hertz. The authors also did a control that they didn't see this phase locking activity during baseline when there was no stimulation in the Purkinje cells. They also looked at a different DCN cell to make sure that only the ones that were inputting to this DCN cell were showing activity, and they didn't see any phase locking in other DCN cells, so only the ones they're interested in. That's good. And then you can see the similar pattern as what you see in figure D, that as the spiking activity, or the percentage of the spiking activity is clustering around the intervals of the interspike interval. So you can see these patterns here. These are for the same firing rate. So the orange is for 40 hertz, green is for 50, and the purple is for 100 hertz. And what you can see is the same patterns of activity for in vitro and in vivo. That as, let's say for example, the 100 hertz, that most of the activity will happen every 10 milliseconds. And then in the, you can see that in vivo and also in vitro, that every 10, the clustering of the spike activity every 10 milliseconds. The authors only showed, on the right, they showed for 50% synchrony. I think this is, on the left, they didn't know what synchrony it is because it's in vivo. So they compared the results from the in vivo to the different in vitro patterns that they saw. Even though in the paper they only provided the right one, which was 50%. What they concluded is that the in vivo results are most similar to synchronization in vitro around 10 to 15%. So even though they only showed 50, the results you see on the left are probably closest to 10 or 25% synchronicity. So what does this all mean, I think is the question. So Mickey touched on this earlier, but if Purkinje cells are inhibitory with this really high firing rate, then how do these DN cells fire? When will they fire? So the previous model was that you have these pauses in Purkinje cell firings. That whenever the Purkinje cells are not firing, that's when the DCN cells will fire. And then you needed this 100 millisecond delay for this to happen, even though there were some questions that maybe this isn't actually good enough for an actual animal to exhibit fine motor control. So what the authors are proposing instead is that there's time locking as an alternative. That the DCN will fire only when there is synchrony in its inputs, and that has nothing to do with its time gap between Purkinje cell firing and DCN firing, but it's about synchrony and not these gaps. Some of the factors that they identify were the fractal synchrony, what percent synchrony needs to be there, is it 10% or 2%, the precision, how well the timing matchups, and whether any excitation is a factor at all. They didn't really address excitation in this model, but the DCN cells do receive some excitatory input even though it is weak, so they want to know a little bit about that for future research. And additionally, they're interested in, for future research, about how DCN spikes are processed downstream. So the red nucleus is the output, and receives the input from the DCN cells, and they're wondering what kind of summation will occur there, but the authors don't address that in this paper, but they save it for future research. And that's all, if you have any questions. Cool. All right.
How much time do I have?
No, it's
That's okay.
what do you plan for?
25 minutes.
No.
Hello, my name is Johannes, and I'm presenting the second cerebellum paper, links from complex spikes to local plasticity, and motor learning in the cerebellum of awake behaving monkeys, by Medina and Lisberger, Nature Neuroscience 2008. And first, short recap of cell types in the cerebellum. We just saw it in Yoni's talk. I'll be talking mostly about climbing fibers and Purkinje cells. Purkinje cells deliver the main output of the cerebellum. And we also saw in the lecture that Purkinje cells have two different types of spikes. that are called simple spikes, here in the red trace or here in the gray trace, but they also have another type of spikes called the complex spikes, which look like this in a recording from slices. And the paper is about a mechanism for motor learning. I think it's quite interesting because they propose an entire system of how errors that you make in a certain motor behavior lead to changes in the cerebellum, and how this is transmitted by complex spikes. So the system they propose is that whenever you make a movement error, then climbing fiber, let me go back, climbing fiber is activated, and that this is some kind of error signal to the Purkinje cells, that then the Purkinje cells change their simple spiking, and that this change of simple spiking leads to a different motor behavior. And the idea of the paper is to test all these things in one single experiment. So what is the idea? They work with rhesus monkeys and record from Purkinje cells in the floccular complex. The floccular complex, this is the inferior surface of the cerebellum. Oh, it's difficult to see. So it's somewhere here. I don't know more about it. And these the cells in the floccular complex are interesting because they have a preferred eye movement direction. So whenever the rhesus monkeys move their eyes into one specific direction for one Purkinje cell, it fires more than when it moves the eyes in the opposite direction. So the idea is to record from these cells and make the monkeys perform a motor learning task involving eye movements. And then the question is, if complex spikes and simple spikes in these Purkinje cells reflect the motor learning in the eye movement task. So how do they do it specifically? So first they find a Purkinje cell in the floccular complex and determine its on direction. So the monkey has to look in all different directions, and they measure the firing rate of the Purkinje cell. Then the monkey has to follow a target, or I can show it here. So this is the screen that the monkey looks at, and in the learning trials the target moves in one direction and then changes direction and moves into a different direction, and the task of the monkey is to follow with its eyes this dot on the screen, and it gets a reward only if the eyes stay within a very small window around the moving dot during the whole trial. The monkey had to fixate and track a light beam on a screen. And it moved for 250 milliseconds in the one direction, and then for another 450 milliseconds in the other direction. And then it received the reward, as I said. So
it was always these 250, so the monkey knew when the change was going to
So
happen?
the idea is that the monkey learns that there will be a change in direction, and that it somehow anticipates with its eye movement this change. And actually you can see this. So here these are the learning trials, and plotted here is the horizontal eye position and vertical eye position. So here you see that after some time there's a change in the vertical eye position, which reflects that the monkey tracks the movement here. And the left panel are the learning trials, and then after the learning, after 100 trials, they randomly inserted probe trials. I think every 10th trial, but with some jitter, is probe trial. And the probe trial means that the target doesn't follow its trajectory from before, but just goes straight. And you can see that before the learning, the vertical eye position doesn't change much. It stays more or less at 0. But after the learning, even if the target moves in this direction, the vertical eye position goes up, and then the monkey realizes something is wrong and goes down again. So here you can see that the monkey has learned to track a target that moves in this direction, and even if the target doesn't move in this direction, the eyes still make this small jump to anticipate the target movement. So you can see that the learning task actually did something to the eye movements of the monkeys. The behavior is there.
Prediction.
Yeah.
The word of faith.
Right. So now let's look at one Purkinje cell and the simple spike firing rate of one Purkinje cell. I'll first explain this plot. Here you see again the movement of the dot on the screen. This time it's a different direction because they always or we now always look at what they call off learning. It means that the monkey learns to move the eyes out of the preferred direction of the Purkinje cells. This line here is the preferred direction. So if it moves the eyes in this direction, the firing rate is maximal, but it learns to move the eyes out of the preferred direction of the Purkinje cells. This is what they looked at. Can't tell you the reason. And here this is the probe trial again. So these are the rare trials where they deviate from this trajectory to test what the learning did. And then in the other panel you can see the results. In light blue you see the eye velocity, and here the simple spike firing rate of one Purkinje cell before learning, and then the red trace is in the probe trials. So you can see that the eye velocity is changed very much with respect to pre-learning, and also the firing rate of this specific Purkinje cell is changed very much with respect to before learning. So it looks like that in this specific Purkinje cell, the spike rate change and the eye velocity change align very well. Okay, and the on trials, I ignore them because it would be too much. There you have not a decrease of firing rate of the Purkinje cell, but an increase. But I won't mention it anymore. So from this, they asked how well do the firing rate changes and eye velocity changes align. And so they looked at all Purkinje cells together. And here, let's look only at the off learning, which means out of the direction of the preferred direction. And you can see here the average of the firing rate changes of all Purkinje cells in the red trace, and the black trace is the eye velocity. And here you can see that there's a very good alignment between the changes in simple spiking and the changes in eye velocity. So they call it Purkinje cells model eye velocity. And you can also see that there's a slight anticipation. So the firing rate changes anticipate a little bit the eye velocity changes. Or you could also say that the eyes follow the simple spiking of the Purkinje cell. And the left panel is 45 out of the 66 Purkinje cells they recorded from. And the right panel, they had another 11 cells that don't model the eye velocity but the eye acceleration. And they call it group 2, but I will also ignore them because it becomes complicated. So far they have shown that monkeys can learn in an eye movement task, and that the simple spiking of Purkinje cells and the learned eye movement align very well, and that the eye movement lags a little bit behind to the firing rate of the Purkinje cells. And here, this is the position, the time point where the target changed its direction. You can see that there's a clear anticipation. So the firing rate as well as the eye movement changed before the actual change of direction in the target that they had to track, which is learning. Okay, so let's go to the complex spikes. At this point, they ask how do the Purkinje cells change their simple spike firing rate. And the idea is that every complex spike induces a change in the simple spiking afterwards. So now they look at, during learning, are there more complex spikes than outside of learning? here you see the probability of complex spikes over the time course of the experiment. This is the time point where the target that they have to track changes direction. And this is the probability. On the y-axis you see the probability of complex spikes. Let's look only at off-learning. You can see that shortly after the change of motion direction of the target, the probability of complex spikes increases very much. So the probability goes from 0.1 to 0.5. Whenever there is something to do for the monkey. So at this point the monkey sees, okay, the target changes motion, so I have to change the eye direction, and at the same point there's a complex spike. Okay, and here they said, okay, the probability goes up only to 0.5, why isn't it 1? And they asked whether maybe there's a variability in eye movement from trial to trial that explains the occurrence of complex spikes, but no, they didn't see any correlation between individual errors and individual complex spikes here.
Why
Okay.
would you expect 100%?
Pardon?
Why would you expect 100%?
No, I wouldn't expect it, but they asked, and maybe one would expect 100%, or more likely one would expect that whenever the monkey makes a bigger error, there's a higher likelihood of a complex spike. But that's not true. So there's just, or at least they didn't find it. So, but now the question is that is this. Let's look at individual Purkinje cells and look at how much their complex spike probability is changed, and then let's look at whether the simple spikes also change more. So the idea would be that complex spikes are an error signal, so there should be a bigger difference in simple spikes whenever there are more complex spikes. And this is this plot. I would like to concentrate on this area here, the filled red dots. Each dot is one Purkinje cell, and on the x-axis you have the complex spike probability in percent of baseline, and on the y-axis you have the change in simple spike activity. And you can see that for the filled red dots there's a huge increase of complex spike probability versus baseline, and also a big decrease in simple spike activity. And this is also, it's a good correlation, good negative correlation. So it means that at least for the filled red dots, more complex spikes lead to a bigger decrease in simple spike activity, which makes sense according to their model. But then, if we look at the filled blue dots, the axes are still the same. You see that the complex spike probability is scattered somewhat around baseline. Baseline is here, but still they have increases in firing rate quite dramatically. So the authors conclude from this that complex spikes are not the only plasticity mechanism, that there must be something apart from complex spikes, because complex spikes don't explain this plasticity for the blue dots. Any questions? No? Okay. So one more thing they look at is the speed of learning. It's not very interesting. So here you see sets of 10 trials. So these are the first 10, 10 to 20, 20 to 30, and so on. And you can see that basically only in the first 30 trials, the eye velocity here or the simple spike response change. At least in here group 1, these are the velocity modelers. Let's not look at group 2. But the complex spike probability, which you can see here, stays above baseline even after 50 trials. So it's remarkable that after 30 trials there's such a huge difference in eye velocity and simple spike response, and this doesn't change anymore even though there are complex spikes. it doesn't fit 100% to what they wanted. Okay, and now after all these correlations, they come to what they think is their most important point. They look at individual trials, individual pairs of trials. In the first trial of the pair, there's a complex spike, and in the second trial of the pair, there's no complex spike, or there's no complex spike in either, and they want to see whether there's a bigger difference in simple spiking in those pairs where there is a complex spike in the first trial. This is a bit complicated, so they call it 0-0 trials and 1-0 trials. So 0-0 means no complex spike, no complex spike. 1-0 means complex spike, but subsequently no complex spike. And they want to see whether the simple spiking changes more in these trials than in these trials, because complex spikes are supposed to be a mechanism of plasticity. And this is the result. It's a bit ugly. This is the time again, and here you see the difference on the y-axis, difference between subsequent trials in simple spike firing rates. The blue line is for the 0-0 trials, and the red line is for the 1-0 trials. So what it basically means is when there's no complex spike in the first and subsequent trial, the change in simple spike firing stays around 0, but when there is a complex spike, and I look at the simple spikes in the next trial where there's no complex spike, then this is the red line, there's a decrease in simple spike firing. And the black line is for statistical comparison, where they just randomly chose, I think, 200 trials without looking whether they are 0-0, 1-0, or whatever. And there you can see that the presence of a complex spike doesn't predict any changes in simple spiking.
What is the x-axis? When is it? I mean, is it after the two trials or in the second trial?
It's averaged about all of these pairs. This time axis?
Yeah. So
this is the time course of the motion of the target. So it starts moving at 0, and at 250 it changes direction.
But in which one in the second trial?
In all trials are the same. All trials are a moving target that changes direction after 250 milliseconds.
No, but it's in the second trial.
In the second, so they're looking at pairs. So this is the second trial. What happens in the second trial?
Yeah, no, they are right. Yes, it's true. Yeah. Or you could say that they are looking at the difference between simple spiking so it's the second trial minus the first trial in simple spiking. And it makes sense to look at these differences in each time point because the time course of each trial is exactly the same. So for each time point they looked at the simple spike difference, first trial minus second trial. Yes. So and here they say that so I said that the target changes direction at 250 milliseconds, and they say that this change in simple spiking is also most prominent here in the most important time window. But I thought it's also quite prominent here, and this time window is not relevant. So I wouldn't be too sure about it. Okay, and now we can summarize. Looking at Purkinje cells in the floccular complex, their simple spike firing rates change in an eye movement learning task, and the eye velocity follows the simple spiking of Purkinje cells. Purkinje cells fire complex spikes at critical times for learning, and after complex spikes, the simple spiking is depressed. overall it fits quite nicely to the model that they proposed in the beginning. There are some things that can be discussed. So complex spikes can be seen as an error signal. I think there's not much to say about it. From other experiments, it seemed already clear that complex spikes cause depression at synapses that were active around 150 milliseconds before the complex spike. And they also confirmed this here. But they also saw Purkinje cells that increased their firing, which is not compatible with this idea of depression. So there must be something more complex going on. Which they also state, this is not the only plasticity mechanism in the cerebellum. And then one objection that one could have is one could say, okay, maybe the Purkinje cells just follow the eye movement. Maybe the monkey learns to move its eyes in some completely different part of the brain, and then the Purkinje cells in the cerebellum just follow this eye movement. But this is very unlikely because stimulation in the floccular complex produces smooth eye movements, and because the change in simple spike firing is earlier in time than the change in eye movement. And that's it. I have no thank you slide, but thank you.
Question? Okay, so we didn't actually go through all these models of plasticity in the cerebellum, which is a bit unfortunate. But the two things that I wanted to highlight here. One is that this idea of this very strange Purkinje cell that gets hundreds of thousands of synaptic input from the parallel fibers, and then only one timing fiber, which is so strong. This was kind of caused many people to believe, even before physiology, that there's something like a teacher mechanism, that the climbing fiber is doing something like a teacher that gives a signal that is very strong that would induce plasticity. And in fact, it was the first, I think it was the first place where synaptic depression, LTD, has been shown by Ito in the early 70s between the parallel fiber and the Purkinje cell. But what is actually very interesting about this work, and especially the last figure, is that usually if you look at protocols of plasticity, what people would do is record from a Purkinje cell and then stimulate a climbing fiber something like 15, 20, 100 times, and then measure the change in the plasticity of the parallel fibers onto the Purkinje cell. So this is not like one activation, it's hundreds of them. And this is actually kind of, in a way, courageous even to look at what happens between trial to trial. This is, if you would do a slice experiment, I think no one would trust that, you give one timing fiber and you see plasticity. So this is interesting because it's in vivo, during learning, and they're looking at one kind of, it's obviously averaged over many pairs, but the effect of one complex spike and the fact that they find statistical significant differences doesn't mean that it's true. It's because it's, as Johannes have shown, you could make the same claim about other places. So statistics here is obviously very dodgy, but it's interesting, at least the idea that you can actually look for it and even pretend to find it is interesting. All right, so let's take a break, and we have a very long paper afterwards that we'll have to do pretty quickly. I don't know if we'll manage to do all of it, but let's be back on time.
זה בדיוק העניין, כאילו ממש אתה לא יכול להוכיח.
אז זה מה שאני אכתוב בכל
מנסה לקרוא תוך כדי
מבחן
כמה
סיום
רגע,
בבדיקות.
מה זה קלפים?
יש לי שאלה, אז אני
מה
אכתוב,
זה?
אין לי אף
כי התחלתי לקרוא כששאלתי את ההודעה, ואז אמרתי אוי, לא. אז אמרתי אוי,
וואי, המאמר הזה, אני לא יודע, לא הבנתי.
אז לא כתבתי לו
לא, גם לא.
כן.
הגרף בסדר, אבל אני צריך להבין את הדברים האלה, מה זה
זה פשוט העמוד הראשון שכתבתי על אחוז הטרנספורטים.
פשוט אומר שהם זה פשוט לא, הבעיה שלי עם המושגים, האורתוקינטי, לא הבנתי בדיוק איך הוא עובד.
אני חושבת שזה
לא, כי מה שהבנתי, כי יש לך משהו שהוא גם נע, אבל גם כמו הראשונים שיש לו
אז איזה רציטוקים מסתבר שכאן
העיניים ואז שזה כאילו
רואה מה שכאן האנליזה היא יותר חדשה. זה הכוונה.
שיש בה
פשוט צריך לעקוב אחרי זה עם העיניים.
לא, זה איזשהו רפלקס שעושה לי קורקציה. אני לא יודע בדיוק מה זה, אבל גם בגלל זה
נכון?
הקטע הזה.
ויש גם אחרי זה דיבילות.
אני לא הגעתי לקודם של האמצע.
שהכל ברור.
כאילו,
סוג של רפלקסים.
נכון, הם לא מסבירים.
היה מקום שהם לא הסבירו.
לא, אבל היה כזה עם הדברים שקוראים באוזניים.
נכון, לא
אוזניים,
זה מוזר, שהם עשו ככה בעבודה.
זה הגרוי.
אז למה הם נתנו לו גרוע איפה?
דיברו עכשיו שזה...
לא, זה נראה לי שהוא לא יודע כאילו מה לעשות.
בדיוק, אבל לא הגרוע, מה זה?
לא,
היית בטוחה שמגיע, לא תמיד
לא, זה אותו דיבור.
דיברו באיזושהי מניפולציה
זה הכל שעשית אקס.
שהנוירונים יהיו קשורים בין אחד לאחד ולא פעם.
לא, אבל אני לא יודע, זה נראה לי בעיניים ובאוזניים.
ואז כאילו אומרים, אם עכשיו אני אעביר משהו ככה, וזה אם אני מבין הסינוס שיש לך, אני לא מבין מה
כן, זה כאילו מה שנראה.
כן, אבל הבלבול שלו בא לידי ביטוי בעיניים.
למה צריך להבין למה אחר?
vertical artifactive.
מה?
כן, אבל משום מה שאומרים vertical.
אבל הקשרים האלה זה קשרים כאילו...
לא, נכון, זה בגלל שיש.
כל הזמן פועל בעולם.
לא, אם אני מבין את הלוגיקה פה,
לא מגיע לאדם.
אבל אין להגיד, הוא בחוק מאוד יש כל
אם בכלב מפה הוא
לאוכל.
אם יש מישהו שיעשה מפה הוא
מה שלו,
זה ממש מדי.
מה שקורה פה,
אבל הנה אצל אנשים.
התגובה לפה בשביל מישהו שמגיב לפה צריך ללכת התגובה לפה בשביל מישהו שמגיב זה מה שאני מבין,
שמה?
צריך ללמוד 180 מעלות.
זה לא כדי כך.
יש באמת...
זה לא מתייחסים יותר מדי
גם
להתנהגות.
שם זה מגיע לעין.
אמרו שאני למשל יותר גרוע מאשר אמר שאין לך באמת.
לא, איפה ראיתי את זה?
קונטקסט
לא, איפה? תגידי לי איפה זה?
זוכרת?
לא
אמרתי לי אחת
אבל זה לא 180 מעלות הפרש, זה בין 100 ל-160. לא יודע.
באייזנקר? כאילו כן, אז כתבו פה איזשהו על עכברים עם בלי טטרופין. בדיוק, פה, כאילו, תציג את זה.
Which like protein אצל העובדים זה מה יש לך significantly worse. אלה אבוקה נשארים?
איפה?
זה העמוד
Only one on one, but it didn't get much. It's on one.
בימים שיש לנו קונדנסים עדיף לא לעשות הפסקה.
לדעתי, יש לך המון קשה רק בגלל שכאילו...
יש פה בזמן הדיון?
כן.
זה צריך להסביר את מה שאתה
כן
אומר.
ולא, זה ממש בסרט, אבל...
בסדר, עדיף לא לעשות הפסקה באף שיעור. אבל צריך הפסקה.
לא, כלומר... רק בשביל האנשים שעושים את האפליקציה הזאת.
אני גם צריך הפסקה, יש לי יושב 5 דקות.
לא, כאילו, אם זה היה קטנה, יושבים שם מדברים 2.5 שעות. אם מישהו צריך, כאילו...
אם יש, כרגע...
לא, פשוט...
כנס הזה, cell death or whatever, זה בשכונת שאננים. אני יודע שאין פיזיולוגיה ביום חמישי.
Yeah.
I don't know.
Shows גם.
You sure?
Right?
But I could certainly see Chaim like,
Oh, he's going to be there.
but I could see him sending Ron to do the.
I can't even go to the restaurant today because Stan Tane's going to shoot. In camp.
כמו שראינו ב...
Stan Tane, Jeff Litzman, Rainbow.
Listening.
Those Tane and Litzman are the team that came up with the rainbow. So you can't even talk at 12 o'clock in camp. It's going to be a great time. And the worst thing about these camp talks, they're not recorded. So if you miss them, that's it.
אגב,
Have
מה
you
קרה
gone to
לכם
any of
משהו
these camp talks?
עם...
The fifth floor in Winston? You've never been to a camp?
That's how you broke your arm.
כן, רצו כאילו להשאיר את החלק הוויזואלי,
Is there something from you and my dad?
The quote. Is it cool?
Right now? Oh, okay. I was worried for a second.
ממש דיבר על זה.
No, but it's really, it's really the top secret. When they're at it. Like, the last one I went to was someone who created like a cartoon show. Like that sold like whatever millions of dollars. Really good stuff.
The DLP tour started twice, so it's not
של מה?
Tomorrow. But I'm talking about Thursday.
Thursday physiology is cancelled, and then there's
דווקא המפליטות זה לא פה.
למידה.
יכולה כבר להתחיל לשנות.
How did you get the exact time? Where you find the schedule? Like the
They
exact
think,
schedule online?
yeah, they schedule. I just added it in my calendar.
I don't remember if I actually registered for that. Even though
I
like sent me
registered.
1,000 emails to register, and I'm like, if I registered, why are you sending to me again?
I know, they don't send you a confirmation email when you register, which I think is stupid.
Yeah, that's what makes the register.
They don't send you a confirmation email. They get your email, but they don't send you a confirmation. What's it called again? Alta
Cell death.
BLTC.
BLB.
You know, BLDT is also my favorite type of sandwich. Bacon, lettuce, bacon, and tomato. BLT is bacon, lettuce, and tomato. That's a famous sandwich. Yes, that's true.
Ah, here, yeah. So the 7 o'clock.
Jacob, do you know what a BLT is? BLT?
זה עניין של
Okay, this is a culture thing. No, BLT is an American sandwich. Bacon, lettuce, and tomato.
Yeah. I heard that.
I made a joke, but I don't know. Maybe initially don't know what a BLT is. So like,
In your presentation?
no, just now.
Okay.
Because the conference tomorrow is BLTT.
And then the
So
Friday.
like it's bacon, lettuce, bacon, and tomato. But it was, I think it's, that one didn't
Friday
translate out.
מה זה?
Huh?
לשם שישתמשו על מה?
I think it will be good to see.
I'm not going to come.
I just looked at it.
It looks good actually.
I think
Did
we
you
will
get
never
recording
miss
and
the
simulation?
picture.
I hope.
It's day one.
Right, and then we get rebonding and stuff.
It's been one, I know.
נכון? שאתה היית
Oh no, this is actually the best stuff.
בחזרה.
This guy, Dimitri Rydberg. This is the best.
נכון, he's coming.
I would like to, but time.
He was at Janelle's farm, and now he's at, I think, Rutgers or Duke or something. And
Rutgers?
he's like a genius in terms of
זה עדיין שייך
what's it called? Microscope design.
לבד ניצבי, או נראה פרספרשן, זה ביחד? כן, זה נראה לי פרספרשן שהוא גם חלק מהעניין.
It's like all labs in Canada, honestly, like just pump out matrix papers. His lab is special, and him along with
קשה לקרוא את זה בלי
what's it called?
וואי, כמה שאני הסתבכתי עם זה, כי הנקודות
Him along with
גם עם צבע. לא, אני דיברתי עם שעתיים להבין מה הנקודות האלה אומרות,
it's not microscopy
ספייס
by
של
itself.
הסימפל ספייס לא כמעט מעוות בין אור
Hello?
לחושך, לפי הפרדיגמה. זה
דיטקשן
כאילו,
by
הפאזה לא
כן.
משנה במובן, אתם רואים שיש עוד הבדל של 12 מעלות.
יופי,
לפי הפרספקטיבה מנסה מלא פרוסים ומשהו.
אני לא יודעת,
עם תעודת זהות אתה צריך לבוא?
כאילו, יש לזה אפקט על הרחבה בפריבילגיה.
נכון, כן, כן.
It's
בסדר, תודה רבה. ביי ביי. Svoboda is obviously an expert in two photon microscopy.
He's making his own microscope.
Yeah, it's true, but it's kind of the real expert is there. With them. Svoboda was working with him. He's good, but I think with the breakthrough coming through them, not through Svoboda. He's kind of improving microscopes here and there. It's not like modern microscopy today. It's not like we studied with him on. It's not like rocket science. The basic principles are known. We can kind of tweak it here, tweak it there. So everybody is doing it. So what?
To improve resolution.
To improve rate, mainly to improve the rate, to improve the depth. How deep you can go. That's the kind of the main issues. The best in class is but there are others. Yes, well, I don't know when time to the the other guy is Fritjof Helmchen. Ben? Yeah. Fritjof Helmchen is the other guy. And there's another one which is
So did you hear anybody olfactory coding? Like one of the leaders of the field?
is what?
He's a leader of the field.
I've never seen the episode.
It's not that, it's good, but it's not like leader. No, it's not a leader. He has a couple of good papers. He was engineering. I personally, what? Why did you say that? It's good. It's good, and it's kind of he a little bit pushed the envelope towards behavior, genetic, stuff like that. But it's certainly not the only one, and certainly not. I wouldn't say the leader.
It was really fast.
But he has created new methods. That's why he's presenting at this conference. He has created new methods combining.
Like I'm presenting at a conference, and I haven't created anything. I have to think what it was.
Technically, that's not correct.
Kind of what this guy. No, much more. He has his spot on the sofa. You can see it. His spot on the sofa. Because he calculated the air and the
Yeah, that's basically the same mechanism why I want to use the
It should be in here. But
It should have been here.
I don't think anyone was in here.
Where, when, why?
The inter-lab meeting, whatever it's called. Three-lab, the tri-lab meeting.
The Sunday,
What about it?
It should have been in here maybe. It was pretty crowded.
it's always
Good that
crowded.
it's good.
But the food is more easily accessible.
That's true. Maybe like bring all the tables into here.
Maybe prepare sandwiches.
הם עושים מעגל גדול, אתם צריכים לדבר את זה בהפסקה, אני בעד, אבל גם ככה if you want to make it critical, you have to take care of it during break time. You can't do everything. Okay, one more. Okay, so I just want to remind you that next week we have the Israeli Society for Neuroscience. don't have a class on Tuesday.
Also this week.
And
And also this Thursday.
sorry, so let's begin. This Thursday we have the ELSEC conference in Mishkanot Shananim. And so we don't have a class. And then on Tuesday we have the Israeli Society for Neuroscience. Again, we won't have a class. And then after that, I think we're starting with Father Ganzia, with Chagai Bergman, if I'm not mistaken. I kind of updated the website, so except I think for one presentation of Yifat, everything is on there, including the papers for motor cortex discussion. And I also will put papers for the basal ganglia maybe today. Okay, so this paper by Dowd. Everybody ready? Managed to go through it. I don't think we'll have time to go through everything. But let's start. So who wants to start describing what's going on here?
So they did a genetic modification in only superior olive cells, inferior olive cells, that allowed the climbing fibers that would normally cross the midline during development. The genetic modification removed the factor that allowed them to cross, so they stayed on the ipsilateral side. So that's the red line, which shows from the olive into the ipsilateral side, whereas the blue line on the left side shows crossing.
Right. So everybody's kind of fine with the circuit, kind of understand what's going on? All right, good. What do you think about this message?
What?
What do you think about this method?
This what?
The method. The method.
You know, they do a button chain, so they spend a lot of time to just to prove that everything else
Yeah, we're going
not
to
changed.
talk about that right now. But the idea is like it's just like unbelievable that you can come to something like that. Okay.
And
Yeah. But
no, I was just gonna say that the Purkinje fibers were not different, obviously, and that's the key that they want to show.
the parallel fibers, you mean, or the
And Purkinje cells.
okay. Right. So Matan. This is for you. Some of it. I mean, I don't think we have to go through all the details. But what do they want to show you? I mean, I think you said it very, very clearly. But let's take some examples.
The basic method is to do anterograde tracing. Retrograde, sorry. So they put some tracer on the end of the cell, and it goes back to the cell before the host cell, you know.
Recent. So they inject in the flocculus, right?
Yeah.
In the cerebellum. And this is going back in the axons to the original cell.
So
I mean, we're not in the stage where we can do slides like that and actually see the fibers going through, right? I mean, you know that there is a method to do that today, right? Or not? Who knows about that? About the method to do that. I mean, ideally what we want is to look at the whole brain and look at the cell and see that the fibers are going through, you know.
DTI.
DTI is possible, but I don't think you'll see it. It's a mouse. You know, the whole cerebellum is like 3 pixels. It's a bit difficult. So what is a method?
I mean, isn't recently there was development where the guy makes a clear brain?
Right, the clarity. Have you heard about clarity? Okay, right. So we can now. I mean, we cannot people can, and it takes few months, but you can, make the whole brain clear. And then basically you would be able to do that, which is also amazing. But for now, or at least this has been published this year, it wasn't available yet, so you have to use other ideas, right? So you inject the virus here, it goes back, right? And then you look for the original location where it comes from. So this is that, right?
In this time you can see that if you inject whether the tracor on the left hemisphere goes to the right hemisphere in the control, but in the knocked out mice it goes to the also to the ipsilateral hemisphere, but also for the contralateral. You can see it by percentage on the columns. You can see with the dorsal cap and the long abrogation or something. You can see only in the contralateral in the control, but in the knockout it's mostly in the ipsilateral, but also in the contralateral. And through the paper we keep referring to this kind of to the residue of this process. But one of the control, not the control group, but the control disease, is just to see that the other parameter didn't change. So the area and the area of the neuron or the number of neurons stay the same for the knocked out and the wild type. This is what you can see in the rightmost panel of this.
Right. Okay, so yeah, indeed, so they kind of you can imagine that when you do something so obscure on one hand and on the other hand so big, the major concern that you have is that any phenomena that you will see could be due to many other things that have gone wrong. So they went into a lot of work to prove that this is very specific, and this is part of that. And then we have the other part. Okay. So what's the other thing that they was what was the other thing that they were worried about?
They wanted to see that the Murphy fibers were not affected in the same way that the climbing fibers were.
Right.
So they checked that, like in their projection to the ipsilateral and the contralateral stay the same in the control and in the mutant mouse.
Right. So this is again a lot of immunohistochemistry work to say that this is kind of it's very technical. I don't think there's much point in getting into all the details of that, but that's the main that's the bottom line. Okay. What's that? Who wants to present that, please? Anyone?
The sigh of relief when you see supplementary.
It's just a combination, yeah.
Yeah, it's a supplementary figure. I put this one because it's important, because it touches upon something that we haven't talked about at all when we talked about the cerebellum and in general. What you see here is a Purkinje cell, right? The green one is a Purkinje cell, and the red one is a climbing fiber, kind of making the boutons on the. You can see the dendrites very well. I think they're actually going down. You can see them very well, but the climbing fiber boutons on the Purkinje cell. Okay. So have you ever asked yourself how come we have like one climbing fiber per Purkinje cell? How does it work? And usually these things work in neuroscience. Similar, I think I mentioned that when we talked about muscle, that we have one, when we talked about motor units, that each muscle fiber is in a innervated only by one motor neuron. And I told you that there is a competition there. There are a lot of fibers coming in, synapses, and then they are doing like a competition. So this is the other classical system for synaptic competition. So at the beginning, each Purkinje cell, like few days after birth, every Purkinje cell is innervated actually by more than one climbing fiber, usually up to I think 6 or something like that. And then there is a competition that we don't really understand yet the details of that. But there is a competition where there are synapses eliminated, and eventually all the climbing fibers are gone except one that is staying, and this will be the climbing fiber of that Purkinje cell. So what you see here is in control, they do this protocol where they stimulate one climbing fiber and they get an EPS, they get a strong EPSC, so current going into the cell. This is voltage clamp. So the current that goes down is an excitatory current going into the cell. Okay, and then you see that the other pulse, the second climbing fiber spike is translated into a smaller postsynaptic current. And this is because you have synaptic depression. So this is a classical case where you have the second response is smaller than the first. And what that means is that you cannot stimulate a single fiber. You don't know where it is. You cannot stimulate in the olive. So you stick the electrode in the slide and make pulses until you hit something that will make you will give you this response. And then you make it smaller and smaller and smaller until it's like the minimum that you will get to get a response. But then in order to know if you stimulated one or more, you want to know that you have only one climbing fiber. Then you give another one, and if it's actually depressed, it means that you stimulated the same one twice. Okay, so you see that the same thing happens in the knockout mouse, okay? Except in one case where you see two. So you see this is one, and then you see another one. So out of all the ones that they measured, which I think is around 16 or so, or maybe more, they had one case when they had double innervation. So this is like, for them means it's not significant. Again, the system is wired properly. Because you could think that the ones that coming ipsilateral, they will make many more innovation, and the synaptic competition would be screwed up.
Right, how does that even work? You have almost double the amount of input.
What do you mean double the amount?
I mean, with the ipsilateral climbing
Why?
fibers.
No, because the other side also goes to the other side. Yeah? You get actually, olive cells are just going to the same side instead of crossing and going to the other side.
How much climbing fiber, how much Purkinje cells are there per climbing fiber? Just one?
No. There are actually, I don't remember how many, but there are more than one. I think it's in the tens or something. They kind of organize in a very interesting way along the climbing fiber is branching out, and then it's kind of innervating many Purkinje cells.
So in the competition, they don't die. They just choose.
They retract and they choose another one.
Another one.
So usually they kind of make synapses on several Purkinje cells, and others are also making, and then they kind of, some of them, it's a very strange phenomenon, but it works. Some retract, some stay.
But each Purkinje cell gets input from only one?
Yes. One. Okay. And this is another supplementary figure to show that all the cell types and the mostly fiber rosettes are the same as well. So Purkinje cell looks like Purkinje cells, stellate, Golgi cells all look similar. Okay. Okay, back to results. So this is this is also, sorry, this is another supplementary. Okay, let's skip that. That's basically just to show you, but you've seen it now in the papers that were presented. They can actually extracellularly see complex spikes. So this would be a complex spike, we call it extracellularly, and this would be the simple spikes. And you see that after a complex spike, you have a pause, so the cell doesn't fire for some time, and then start to spike. And these are similar So also on the, not only on the anatomical level, also on the physiological level, the cells behave normally. Okay? So the knockout doesn't screw up things. Okay. Results. First result figure. Anyone? Olga?
So they presented sinusoidal visual stimulus in different frequencies. So this is the top graph. And then they plotted the complex spikes. So you see here the pace gauge and the muscle pulse, and you can see that the blue is the control and the red is the mutant. And you can see that the preferred phase for the complex spikes is reversed between the control and mutant.
Okay.
Can you explain more about the optokinetic stimulus and
So what is optokinetics reflex? What does it mean? What is it? Anyone knows what it is?
Moving visual stimulus?
So basically when you have something and it moves, your eye moves with the stimulus. Okay. And this is different than when your head is moving with the stimulus. You can actually track you would track it until at some point you lose the thing and your eyes will jump back to where they were. So that's the reflex.
okay, but the stimulus,
And the stimulus is moving bars. They have like bars and they kind of rotate very slowly. And you kind of and ideally your eye would track the stimulus, will move it with the stimulus. And we'll see in the last figure whether they do that or not. But
you don't measure the reflex.
you don't in this place, in this point, you don't measure the reflex. You just you measure the activity of the neurons.
Does it mean that in one side, all of the complex spikes prefer the same phase of the stimulus? Because otherwise you wouldn't have a shift in the phase. You will have a uniform distribution.
What do you mean on one side? I don't understand.
Their main point is that when the
They don't record from the whole cerebellum. They record from a specific place that is known to, as Johannes, it's exactly the same place that Johannes was talking about, that is known to be responsible for correcting eye movement.
but all of them to the same phase of the stimulus. We don't see in the normal in the control. We don't.
There is some distribution, right? I mean, they don't show it here.
But it's not a uniform distribution. Otherwise, they wouldn't. It's not uniform. Otherwise, they wouldn't have a shift in the phase.
No, yes,
So
yes. They kind of tend to. They tend to like the peaks in control. They tend to synchronize with the peaks of the stimulus, and you see that it's kind of also it's kind of a little bit independent of frequency. So it tracks the stimulus. Whether it goes fast or slow, it kind of tracks the stimulus.
From the control, if we were to recall from the other hemisphere, would you get the same result with the control or the same result with the mutant?
From the other side of the, you
Like
mean from
one
the
hemisphere
from the
prefers
other?
this phase of the stimulus, and the other hemisphere prefers the other phase of the stimulus, and that's why we get the
I
shift.
think it will be like control because the eyes moving together.
So why
And
would
we'll
we
see it later.
So if it's the same, why would you get the shift with the mutants? I thought that now the mutants act like the right hemisphere thinks that it's the left hemisphere or something like that, because the fibers are going into the other.
Let's talk about it in a second. I'm confused myself about that. It's a good point.
But did they
It's
give
the role?
what?
did they give any explanation for why in control the complex spikes are somewhat locked to the peak or falling component of the stimulus?
No, I think it's kind of apparently it's a well-known phenomenon. Kind of as a I know that like optokinetic ZOR and the VVOR, the things that they actually check are almost so they're like two classical paradigms in cerebellum like behavior, cerebellum behavior, physiology, which are these, and the eye blink conditioning. The typical paradigms that people would use. They know where to locate them. One is the flocculus, the other one I forgot where, but there's another location in the cerebellum that is known to be responsible for eye pain conditioning. And these are the like typical paradigms. So it's kind of it's supposed to, if you read this paper, you're supposed to know that in a way. That this is it's not something new for them at least.
It doesn't fit at all with the idea that complex spikes are an error signal or
Right.
a feature, right?
Right.
Has nothing to do with that.
Right, that's true. But it's kind of that's not the only way that people kind of no, that's not the only way that people think about complex spikes. Because you don't encounter error signals all the time, but you do have complex spikes all the time. And it is kind of a mystery what happens, how how the synapses stay stable, as you shown yourself in your presentation, that you have cases where complex spikes still occur, but there's no learning and no plasticity. Right?
And there are cases where the complex spike probability stays the same, but still there's a lot of plasticity.
Right. So it's not one to one. I mean there is something that is kind of okay. Let's not go into. Okay. So now the real question is here. Who wants to present? If we put Ori's question aside for a second, what the main question here now would be?
What will happen to the simple spike?
Right? What will happen to simple species? Because we have two options. One, that they will follow the complex spike. And this would fit with what?
Complex
They are
spike.
routing.
What?
They are wiring.
Well, we know that the motif fibers are not rerouted, right? So why would the simple spike follow the complex spike?
If the climbing fiber is affecting also the simple.
So complex spike
Is
is
the
the
climbing
target.
fiber affecting the simple spike or affecting what?
Because usually
Yes,
the
yes.
cortical.
If the complex spike is influencing the antiphase manner that the complex spike
In
is.
what sense?
If the climbing fibers affect the molecular layer and that cause effect on the power fibers and
Yeah, kind
the
of.
long-term
I'm looking for one word
inhibition.
that we talked about it. What?
Plasticity?
Inhibition.
Plasticity, right? If the complex spikes are inducing plasticity, they will change the synapses that's coming from the climbing fiber. And then we should see correlation with the activity of the complex spike rather than of the simple spike.
You're implying that every complex spike induces plasticity?
No, we're talking now about a very general plasticity. We moved the complex spikes from here to here. Okay, so actually we have a big bunch of complex spike happening in the antiphase, and here they actually occurred during the peak of the oscillation. And now, if they are inducing plasticity on the climbing fiber, on the parallel fibers, we should expect that the simple spike activity would change as well. Okay, this is obviously very. You should realize that this is a very naive claim in a way, in the sense that it could go completely the other way around. I mean, the fact that you rerouted the climbing fiber, okay, means that the Purkinje cell gets different spikes. But it could also mean that the plasticity actually happens at the polyvary neuron and not in the Purkinje cell. So it could be that the simple spikes somehow affect the output that goes to the DCN, that goes to the Olin, and actually somewhere there plasticity occurs, and the spikes are happening on the wrong phase, so to say, because of that. So it's very straightforward argument, but you have to be careful with it because you're looking at an has been there and had to learn to live with its rerouted fibers in general. Okay, so what do we see?
We see also the modulation, the modulation of the stimulus also on the simple spike is the same as in the complex spike. And we see another thing which is a deeper decline of the firing rate in the mutant spike group.
Right. So there is a reduction here in the firing rate of in control, but the reduction on the sideband in the mutant is much stronger. Okay. Right.
So, but how can they be sure that this is actually plasticity and not just this phenomenon that after a complex spike there is a brief phase where no simple spikes can be fired at all?
You mean here or in general
In
here?
both, in both cases. So because it's true, right, that after a complex spike there is a brief pause in firing in general, which is not plasticity, it's just recovery from the complex spike or something. So wouldn't that alone explain this this data?
It's possible. It's completely possible.
No, although this is in seconds, I mean, this is a long time.
Yeah, but
That's
I mean,
true, but
look,
still,
but these also happen during
right.
the same time.
After each complex spike there is a short pause in simple spike firing, and maybe that alone explains the other curve.
It's true.
But then why would there be a rise in simple spike after that?
It's not that you have a baseline and then you just knock it down during this cycle. So it looks as if it goes up. Okay? You would have some baseline and then every time you have a complex spike, you reduce the rate.
But I
Then
don't
it
see it
goes
in the
down.
control. I mean, the control, even though we have complex spikes, we still don't see
No,
shut.
It's not.
Yeah, but it should be a characteristic of the cell in general, not just in the
So yeah, right.
mutant.
What I said doesn't explain why it goes down to zero completely in the
Here.
mutant. Yeah.
No, but let's talk just about that.
Yeah.
Here we have another explanation for that, right?
Yeah.
Ken? Yes.
If you claim that the reason you have decline in the firing rate during specific phase is because of the complex spikes during the same phase, you wouldn't expect that the amount of decline will depend on the number of rate of complex spikes,
Right.
and you don't see that. And
You don't see that
moreover,
why?
you can see that the
Well, what do you mean you don't see that? Why do you don't?
if you compare this plot to the previous one.
But no one told you how much complex.
What do you mean?
They didn't compare
I don't
complex.
understand. So the claim here is that the rate of the complex spike is more or less constant.
Yeah,
Right,
in the
and-
control and in the mutant.
Yeah, forget the mutants, for example. The mutants are complicated. But let's talk about the control.
I'm talking about comparison between the control and the mutant. You can say that you can't compare them, but I'm talking about the comparison.
Okay. No, but Johannes kind of suggests the following. He says, we have an elevation of complex spikes. We
Right.
know that locally complex spikes inhibit the simple spike rate of the neuron. So every time you have a complex spike, you have less simple spikes for a while. So if you have more complex spikes here, okay, then you should expect less simple spikes around this time without assuming any kind of plasticity. And this is what you see here. Okay? And what they claim in the article is that this is a function of plasticity. Okay? And there is one point on that, right, that talks about it in a second. And this is where was it? Here? I think it was here. Right? It was here. We won't have time to go into that, but if you look at this picture, there they're doing they compare the vestibular stationary paradigm in the light and in the dark, right? Where in the other one you wouldn't, and you see that there's no almost very little effect. This is like a transient change in the frequency of complex spike. So you take the mouse and you rotate it in the dark, and you have the manipulation, and then you do it in light. So on top of the vestibular input, you have also the visual input, which induces a lot of complex spikes, because if you don't match them, you would get a lot of complex spikes in addition. And this by itself doesn't change the rate. So they claim that this is not a transient change in the rate of the complex spike, but it's actually a long-term plasticity effect. Okay, a long-term.
Okay.
Okay? Okay, so Pnina, you wanted to say something? I think we'll have to stop with that. But
If, I mean, I said what I had said. I mean, no. Oh, my claim, and I'm still standing behind it, is
Yeah.
that that like if you want to say that the depression of simple spikes is a result of the complex spikes, you would expect that the amount to which it was depressed will depend in the rate of the complex spikes. And here they said that the rate of the complex spikes did not differ between the control and the mutant, and the rate of the simple spikes of the inhibition of simple spikes did differ, and you can see it pretty clearly. So I don't think that even if we have only this result, that you can that this explanation explains the whole picture, that we can see.
Okay.
If you can compare it at all.
No, it's very hard to state because they change the axis.
Right.
It's very helpful.
Okay. Let's see if there was anything interesting. There was one thing that I wanted to highlight, which is more methodological, which is, here so the explanation, right? We don't have time to go into it. The explanation of this sideband is because of plasticity of inhibition. Okay, inhibitory inputs that are activated also very strongly here. So methodological, they record from inhibitory neurons, and they show again that they fire with the oscillation, this is changed with the rerouting. But again, when you do that, you are going into trouble of how do you know that you record from an interneuron? And they give a list of ideas why they think it's an interneuron, but the only method to be able to be sure that this is true is doing that with whole cell recording. Okay, so single cell recording, and then they can compare the properties that they record in the extracellular to the one that they get in the intracellular, but then they can also fill the cell with dye and histologically identify that they recorded from an interneuron and not from an inhibitor. Okay, so let's just go to the final figure and we'll stop there. Okay, who wants to relieve us from our misery? Okay, so what did they want to show here? The punchline, basically.
They wanted to show that motor coordination is adversely affected by having this change in spike firing. And so they show that the gain is greatly reduced in the mutant mice. And they also wanted to show that the phase variance is significantly increased in the mutants compared to the controls, where they say this variance is so small you can't even see it on the graph. And then they wanted to show that the difference in light and dark was that in the dark there was a slight reduction in gain, but the variance was greatly reduced, which shows that this effect is definitely something to do with these light visual signals as opposed to just the inherent visibility.
Right. Okay, and they even make a comment that you can actually compare those mice to mice that don't have at all any output of Purkinje cells, and actually those are doing better because here the signal is actually reversed to what they do. Okay.
What does it mean to have the mice
What?
stable mice? What does it mean? The way you have a leg the change in angle of the eye
It's leading kind of, no?
Yeah.
leading to
Coming
his
in
head.
forward.
How
What?
come?
What do you mean?
Does it predict the movement of the...
It's oscillatory, right? So it's kind of, it's always,
It's like lagging of 200 and something, or...
I think so. Okay. All right. Oh yeah, we'll think about it and I'll give you an answer. Maybe you can come with conclusion.
Can I ask you something about this picture? This one, the universal dark. So, no, it's very space. I mean, the assumption is that in the dark, it doesn't matter how the drum rotates because the mouse
No, I think what they do is that they rotate the mouse
they rotate, but in the dark, the mouse is supposed to be
right. Yeah, so it's only the tip rotates.
so, but I think to me they look quite different darkness fronts. I mean, it's 80 here and 65 here. It's like much sharper.
Yeah, so they don't say there is no difference. They say there's a mild difference, which is accounting for 22% or something like that. If I remember correctly. And this is like kind of, it means that there is some effect, but it's not extremely major. And I think that what they really want to say is when you don't have the visual, still have
Yeah, I'm just confused about the paradigm as a whole because it's always like mice have completely different vision and they see in the dark and it confused me that
what do you mean? No, they don't see in the dark.
they see in light that we don't see. So
Yeah, but when they do, it's actually the opposite, right? No. While, sorry, no, they do see in infrared, but when you do instruments in the dark, you don't have infrared.
yeah.
You do actually, what you usually do is you have infrared of about 940 nanometers, which is then they can see that. I think they see up to 870 or something. So if you use, so we use that sometimes in experiments where you want to see the mouse when it is in the dark. You look at it with 940 and you filming it with a camera, with an infrared camera, but they don't see. Yeah, I think this is kind of cool. So it's not that what in the darkness you see, they don't.
No, but this is what confused me. They do not see in the dark the
טוענת שהיום ברגע
condition in darkness. That's why I was
אבל יש לנו תמונה של...
No, but it's not both in the dark.
no, but here and then this both dark one.
באופן טבעי,
If this is in the
יש
dark,
לך כרגע פוקוס פס רק על דברים שבפיק
it's in
ויש.
this. I don't have a scientific idea
אם הוא
of
כבר
hey,
יורד
if
רק
this is
בפיק
really
של
different or not.
אז למה יש לי פה שינוי בפאזה?
To me,
הנה
it
עכשיו
looks
זה
different. It's
מייצג
better.
את הצד השני
But there is
המקובל.
an
איך הוא עושה דברים
additional
חדשים?
אני עוד פעם, זה לא שאני מבינה את השאלה מה שאת אומרת, אני
One
חושבת
has
שהמושג
the
דווקא
fibula
מראה
the other
אינדיקציה
one
למה
you don't.
שיהיה
אוקיי.
בחזרה.
No, I thought they were certain both
גם בצד
cases.
השני הוא יורד
One they
נכון.
showed the static
אז
drum, and
אין לי
this one
סוג
they
של
showed the
כאילו,
rotating.
אין פה העדפה לכיוונים, כולם
So
עושים אותו
maybe
דבר.
I got it from
זה
100.
פשוט השאלה שלי.
No?
מה זאת אומרת?
It's not a
כל
rotating
הזמן
drum in
נשאר
the
כמו
dark.
שהם
No,
יש להם
that's not the
כיוון
point.
זה מה
I thought this
שיואב
is
אמר במאמר.
static drum rotating
כיוון
drum,
מועדף,
and this
אבל
is just
אין להם
control
כיוון
by
מועדף אם כולם רוצים את
לא,
זה.
אבל הם החליטו רק את אלה שבעלי כיוון
Okay,
מועדף
I got it wrong.
ספציפי.
No, maybe I'm
ורטיקל...
אבל מהם למה כולם יורדים רק בפיק?
כי הם משיגים אחריותם. הם משיגים רק תאים עם כיוון מועדף ספציפי. אלה התאים שמהם הם החליטו. כאילו כתוב לכם משהו, זה נקרא VA for symmetry.
נכון.
אז הם משיגים רק אותם. מה זאת אומרת כולם?
זאת אומרת שהם בחרו רק תאים שיורים פה בפיק. ושיש גם תאים,
כן, הם בחרו רק תאים
אז
בעלי
איך הם בחרו
כיוון
את התאים
מועדף.
פה? הם בחרו בכוונה את התאים שיורים בפאזה ההפוכה?
לא, אז זהו, אז כאילו זה נראה לי... בכל מקרה זה סוג ספציפי של טקסט. זה בגלל.
כן,
תאים בעלי
אבל
כיוון
זה לא הופתע, אז למה, איך זה, כאילו זה...
לא, זה דווקא נראה לי זה הגיוני שבצד השני יכולים לראות הפוך, אבל עדיין את מבלבלת אותם
מה את
ומתבלבלים.
אומרת שהם יראו הפוך, שהם יראו קומות?
שבצד השני הם נראים כמו המושלמים, כן, אבל ברגע שאת עושה להם בלאגן, וגם לא כל, וגם חלק מהצווים זה ממומן.
זה מה שמוזר לי, אם יש להם ציווי מאוד
But
אף,
he
למה
have a
שהם יראו
similar
את
fashion
הצד האחד של הגרוע
he
בצד
don't
השני?
So it's saying
ולמה
that
הם רואים רק בצד העמדות של הגרוע? ולמה בצד השני הם יראו רק בצד השנייה של הגרוע?
here the drum
תמיד
ואני לא
יש לך,
מבינה את
תמיד
המשמעות
יש לך,
של הצד העמדות של הגרוע.
ילד,
כאילו מה שאת אמרת על
can you say
אבל
this
זה
is
רק
rotating
בשמאל.
in the dark
הם יורים, כן, הם יורים רק ב... כאילו כן, יש לך
But then it is
שחור
true
לבן,
that
שחור
it-
לבן, בשני
Yeah,
הדקים,
that's what I was
ואז כל
expecting.
פעם יורים נגיד רק בשחור, או רק בלבן, רק בהערה, כאילו on steps, off steps, כאלה דברים.
אז אם הם רואים גם בצד השני אותו דבר, רק בשחור, אז זה מוזר. שאנחנו בכלל יכולים לעשות איזשהו דיון ללבן.
he's saying because now what
לא, אבל אני הייתי חושבת שיש כאילו on steps ו-off steps, זה שתי השימושים.
when the light
הם
was
לא מצאו
on
כאלה.
כן.
הם מצאו רק את אלה. ואם
so
בצד
the
השני נגיד
vestibular
הפוך, אז אני
now
תוהה מה
by the
בניסוי
orthokinetic
גורם
signal
לזה
was
להיות
moving
ככה
in
שכל
phase
ספירה
with the
תצא
vestibular
אחד
signal
לאחרת שלו.
when the light was on so when the light was off it wasn't moving at all
כאילו תמיד,
so this
כאילו
זה
כאילו
תמיד
זה
שכיוון
הפעל
שינה.
התמיד שהקומפלקס ספייס יורים. זאת אומרת שגם בצד שני,
כן.
זה משתמע
לא,
שזה יכול
נכון,
להיות אותו דבר, אבל כאן זה לא מדמים לי משהו,
בגלל
מדמים לי משהו שהיה קיים.
No, it's just confused about the
אם זה
paradigm.
באמת
It's
דבר
hard
שיש
to
לו,
really
לפני
rush
שמדמים את הצד השני, מדמים משהו שלא קיים בכלל בקונטרול.
כן.
I don't know
לא,
it's
יכול
kind
להיות
of
שגם בצד השני בעצם.
because I think the conclusion from the trigger is that nothing matters
נראה לי בגלל
you know, right?
שניסו באותה פאזה,
כן, כן.
לפי הניסוח שלהם.
But
עד
I
שהם
think
שמו
that
אותם
this is
בדיוק.
control something
אתה
better.
נתן להם משהו מובן לגמרי, שלא קיים בכלל. אז הם לא יכולים להגיד שכל...
לא, בגלל כאילו נגיד הכלים קלאסיים האלה של ה...
לא, אבל אמרנו שהצד השני יודע.
הצד השני אומר אותו דבר, כי הם כאילו הם
אז
יורים
הם לא
כאילו
מדמים
באותה
את הצד
פאזה.
השני כל כך.
לא, הם לא מדמים את הצד השני.
אז
הם
יש
מדמים
לנו
כאילו
עוד
משהו
משהו,
אחר.
אבל יש לנו עוד משהו שהשתנה
אני חושבת שזה
במערכת.
יותר הגיוני. זה לא עוד משהו, זה המשהו שהשתנה. זה המשהו שהשתנה. כאילו את יורה, יש לך תאים שיורים, נגיד, מעדיפים כיוון מסוים, ויורים תמיד בפאזה מסוימת של הגיבוי. שזה הגיוני,